All right. Well, I think we'll go ahead and get started with a few introductions. I'm Claire Weichselbaum. I'm a member of our education and engagement team here at Allen Institute. I'm a neuroscientist by training, but have been here about a year and a half working on our education and engagement team to share all the amazing science that happens at the Allen Institute with educators like yourselves and students, public audiences, and other folks who might find it useful. So, I want to introduce a couple other folks who were involved in developing the resources that we're going to be sharing with you today. So first, I'll pass it to Stacy if you want to just say hi and share what you do here at Allen.
Sure. Hi, I'm Stacy Sorensen. I'm leading the neuroanatomy team in the brain science unit. Um, our group spends all day, every day thinking about morphology, drawing neurons, quantitatively analyzing them to try to understand how their detailed structure fits into a description of multimodal description of cell types. And then we're describing cell type, all the cell types in the mouse brain and now human cortex and some primate. And so we're really passionate about morphology, and it's exciting to see that maybe some of you are as well, or hopefully we can get you there. Anyway, thanks for coming. Thanks, Claire.
Absolutely. Rachel D., do you want to introduce yourself?
Hi, yeah, I'm Rachel Dally. I work on Stacy's team, and I manage the scientific pipeline that enables the generation of these single cell morphologies. Been here a long time and have general knowledge about neuroanatomy, but my heart is definitely in the single cell morphology world. So thanks for being here.
And Ingrid.
Hi, I'm Ingrid Redford. I'm a research associate here on the morphology team, and I work primarily on creating a lot of our reconstructions in human cells and non-human primate cells, and creating the 3D reconstructions similar to the one that you see on the screen.
Awesome. And Rachel P.
Great. Hi, I'm Rachel Penton, and I'll be pretty much managing the chat today. I'm another member of the education and engagement team here at the Allen Institute, and I recently left my teaching faculty position at UNC Chapel Hill to move fully into curriculum development and faculty development. And so my focus is on undergraduate educational resources and faculty development for undergrad educators.
Awesome. And while there are too many of you for you all to introduce yourselves verbally, I'm thrilled that many of you have introduced yourselves in the chat, and we're so happy to have you all here. And let's dive right in. So this is our agenda for today. We're going to start with a little introduction to the Allen Institute and our education and engagement program specifically, share a little about some of the resources that we offer. And then we'll dive into this curriculum that today's workshop is about, our Neurons Beyond the Textbook 2.0. Some of you may be aware that we had a Neurons Beyond the Textbook curriculum before this, and it has been significantly revised and expanded. So that's what we'll be sharing with you today. And then we're going to take you on a virtual lab tour and show you where all of that data was actually collected. So we're going to be walking around the Patch-seq lab, which we'll tell you a little more about what that is. And then have some time for Q&A with Stacy, Rachel, and Ingrid after that as well. And then we're going to move into smaller breakout groups so that you all can really think about how you might apply this to your specific context, whether you might want to implement these lessons, maybe some parts of them, adapt them to your particular situation. We'll have a mural board so you can share your thoughts and hopefully learn from each other a little bit. And then we'll share what was discussed in the groups and wrap up. So we're hoping that by the end of the next hour and a half, you'll be able to describe why you might want your students to be working with open data, like the data used in these lessons. You'll be able to locate the resources we have available on our website, summarize what students will learn in these particular lessons, and explain how the data used in them was collected, as well as start thinking about how you might implement these lessons in your own classes. And I should add, please feel free to put questions, comments, thoughts in the chat throughout. We'll be pausing to take questions at various points, but please just feel free to put them in the chat as they occur to you. So let's start by talking a little bit about the Allen Institute. Some of you are probably more familiar with us than others, but we're an independent research nonprofit established 21 years ago by philanthropist Paul Allen here in the beautiful South Lake Union neighborhood of Seattle. We have about 930 some employees, and we're still growing. And our research is focused on tackling complex, large-scale, foundational questions in the fields of primarily neuroscience, cell biology, and immunology. So you can see here our mission statement is really about understanding life and using that knowledge to advance health. And the Institute operates under a few core principles. So big science, we take on big questions in biology, creating huge, scalable, standardized pipelines that generate these massive data sets, such as some of the data you'll be working with today. We practice team science, so we work in very collaborative, interdisciplinary teams to tackle those big questions. And open science is really fundamental to the way the Allen Institute operates. So we openly share our data, tools, models, protocols, and more with the research community to accelerate impact. And the education team plays a role in creating that impact by partnering with educators like yourselves to share our big team and open science with the next generation of scientists. So we're comprised of multiple institutes within the Allen Institute. The Allen Institute for Brain Science was our first. You may know that we were originally founded as the Allen Institute for Brain Science, but we have expanded beyond that quite a bit. The Allen Institute for Cell Science, which applies the same idea of sort of foundational mapping of the brain to foundational mapping of the human cell, specifically working with induced human pluripotent stem cells. They were founded 10 years ago. Then the Allen Institute for Immunology came on board in 2018, and the Allen Institute for Neural Dynamics, which sort of grew out of the work of brain science to really explore how all the parts of the brain that the Institute for Brain Science has identified work together dynamically to create function, came on board in 2021. Our most recent group is the Seattle Hub for Synthetic Biology, which is focused on using synthetic biology techniques to advance our understanding of development. So really reimagining how we study cells by turning them into their own recording devices, so to speak, using DNA typewriter technology. And lastly, we have the Paul G. Allen Frontiers Group, which funds cutting-edge biomedical research outside of our walls. So that's a quick overview of what the Allen Institute does, the very high-level version. And the education and engagement program really takes all of that science and tries to help educators such as yourselves and others use it to educate, engage, and inspire. So we take the Institute's open data, use it to create lessons, activities, events, and then disseminate those to educators, students, public audiences, and others. So I want to just take a moment to show you where all of our resources are online. All right, you'll see if you scroll down here on our website that you can go to our curriculum and teaching resources. So this is our materials library where you can search. Most of you are undergraduate educators, so you can filter by that and see all of the different resources using our data and tools that we have available. So again, definitely feel free to continue exploring that after today's workshop. So I just wanted to briefly mention some of the neuroscience data sets. We don't have curricular materials for all of these, but we have curricular materials for many of them, and we're currently developing more. So we're probably best known for the original gene expression atlases, which mapped first the mouse brain in terms of gene expression using in situ hybridization, and later human and non-human primate brains as well. And more recently, we've moved toward more single-cell transcriptomics, spatial transcriptomics, and all of that data can be found in the Allen Brain Cell Atlas, or ABC Atlas. The data set that we're going to be focused on today is our multimodal single-cell data that combines morphology, electrophysiology, and transcriptomics collected using the Patch-seq technique, which we'll be talking more about in just a minute. And so we'll be exploring that database a little more shortly. We also have a connectivity atlas, which is axonal projection data, which can be really fun for helping students explore some neuroanatomy and how different parts of the brain are connected. And we do have a lesson related to that. And we also have a massive set of electron microscopy data. So the Microns project, as part of a consortium, has actually mapped an entire cubic millimeter of mouse brain in its entirety. So that's really fun to explore as well. And we also have some physiology data, both electrophysiology and 2-photon calcium imaging, which can be found in the Allen Brain Observatory. And all of these data sets are available on brain-map.org, which is our data portal for the Allen Institute for Brain Science. The Allen Institute for Neural Dynamics has just launched their own website data portal. And so as more of their data starts coming online, we're excited to develop some resources related to that as well. All right. So enough of me talking. I would love to invite you all to share in the chat some of your experiences. So have you ever used any open data in the classroom? Maybe Allen Institute data, maybe data from somewhere else. Share in the chat how that went. And then in a minute, we'll think a little more about what are some of the benefits to students when we do that. So while I'm waiting for folks to type up their thoughts, I will share that we've actually heard of a couple of students ending up on publications from independent research that they've done without ever setting foot in the lab, purely using our data sets. So it's very cool that students can go all the way from a very guided activity, that you might be leading in even an introductory level class, all the way to doing independent research with our data sets. So awesome. Okay, so someone has used electrophysiology data in a computational neuroscience class. So one of the great things about a lot of our data sets is that many of them have really nice online interfaces for exploring the data, including the data sets that we'll be showing you today. But there's, of course, much more you can do with programmatic access to the data. So it can be a really great opportunity to bring in some computational skills. Awesome. Someone has used the connectivity atlas. Okay, someone's done lab exercises and had students design projects. So again, it's great all the way from very guided to more independent research. Using Allen data as part of a cure. Yeah, absolutely. So that's for anyone who's not familiar, that's a course-based undergraduate research experience, which again, it's our hope is that it makes those kinds of experiences more accessible, since you don't need to have the equipment to collect the data yourself. Using open data sets to teach statistics. Awesome. Very cool. And someone has a former student who worked with some of our data in their class and now works at the Allen Institute for Neural Dynamics. So that's very exciting. Someone's used Open Neuro to have students analyze MRI scans. I love that. So MRI is one type of data that we do not have here. So that's a great resource to share. Very cool. Scientific initiation. I'm not sure that I'm familiar with that term. If you want to share a little more about that. Great. So someone has used the older version of this Neurons Beyond the Textbook curriculum module. So I'll be excited to hear what you think about the revised and expanded versions. Yeah, and someone's raising a great point that undergrads aren't always necessarily ready to just be turned loose to explore a huge, potentially overwhelming data set like this. And I think our hope with the lessons that we're presenting today is that they really guide students and scaffold kind of taking those first forays into a large data set like this to help them get ready for more independent exploration. This is great. Okay, well, I'm not going to read every response because it's a wonderful problem that there are too many of them. Someone's used EEG data. That's another great resource. Would love to hear where you're finding that, because that's also not something we do at Allen currently. Very cool. Wow. Okay. Someone has used Allen data to build a model of a whole cortical region. The papers just came out in the last hour, so congratulations. That is extremely exciting, and I'm going to go read your paper after this. Yeah, gene expression mapping and connectivity atlas during COVID. Yeah, so the other nice thing about these resources being available online and accessible with just a computer is that, you know, it works very well for remote teaching. Very cool. Awesome. All right. Well, thank you all so much for sharing. I have one more question. How do you think working with open data benefits student learning, or does it? Authentic, meaningful experience. Yes, and potentially inspirational, inspiring the next generation of research with all the cutting edge tools. I love that. That science is ever changing and that students can be part of discovery too. An opportunity to pursue student-initiated ideas. Yes. Loving all these responses. Yeah, limitations of what you can collect in your own lab shouldn't prevent students from thinking big. It's great. Yep, students need to learn to work with big data also, because that's the future. Yep, working with actual data without the expense and potentially tedium of gathering that data. And it's democratizing. Yes, I love this. All right, please continue to share in the chat. And we're going to keep going just to try to keep us on time. All right, so I'm just going to give you a very quick overview of Neurons Beyond the Textbook 2.0. So as I mentioned, there has been a version of this lesson that's been up on our website for a while, but we've significantly revised and expanded it. So while that original version was sort of a PDF for students to work through, we now have a PowerPoint deck for each of the two lessons, along with instructor notes for you, a tutorial video for the data set that's used in each of the lessons, a guided student worksheet, also with an answer key for you, and an instructor guide kind of summarizing all of these materials. I'll also add that we're going to be creating a video of the lab tour similar to what you're going to experience today in this workshop, so that you'll have an opportunity to show students the behind the scenes a little bit more, and that we really encourage you to view all of these materials as modular, adaptable, take what is useful to you and leave the rest. The student worksheets are going to be editable Word documents. The slides will be PowerPoints that you can remix as much as you need to. All right, so this is one of the data sets that students will be exploring. This is sort of the data set that lesson 1 is focused on. And you can see that each of these sort of cards here represents data from a single cell. So many of them have both electrophysiology recording as well as a morphology reconstruction. And the lesson is focused really on exploring the morphology, specifically how the morphology of real neurons differs from the diagrams that you might find in a textbook, hence the name Neurons Beyond the Textbook. So if we click in here, you can see this reconstruction that you can rotate around. We also have the original images that you can kind of zoom in and out and move around through. So, the worksheet kind of takes students through a little warm-up activity that has them think about their current understanding of a neuron. And then at the end of class, they'll have an opportunity to show their revised understanding after playing with these reconstructions of actual neurons. So students will have a choice of looking at human or mouse neurons, have an opportunity to compare excitatory versus inhibitory neurons and see how their morphology differs, and then think about what that might mean for the function of those neurons, which might be sending information more locally versus further away, which might be involved in integrating more information across cortical layers, and then have an opportunity to work with a partner to see how that might differ across species. So that's the first activity, and the PowerPoint slides are really intended to help you provide any background information that the students will need going into this lesson. So each of the lessons is set up to be a 50-minute class period. You can certainly remix, do a flipped lecture, assign some of the work as pre-work or as homework. But in theory, at least, the material should be able to fit into a 50-minute class period. And by the end, hopefully students will have achieved these learning goals of comparing and contrasting real neurons to a typical textbook diagram, considering how the morphology of the various parts of the neuron might be related to its function, looking at these representations, and then ultimately using the real data to draw some conclusions about excitatory versus inhibitory morphologies. So again, you can browse through these slides on your own time. We kind of take students through the history of neuron morphology a little bit, from Cajal to how we study it at the Allen Institute. Have some opportunities for think, pair, share. And then again, ultimately lead students to this database where they can complete this activity. As I mentioned, there will be a tutorial here that sort of gives a really easy walkthrough of exactly what students are going to need to be able to do to work with that database. So again, this is the student activity portion that walks students through one of our other data sets. Yes, we have a great question about histograms, which I will get to momentarily. So this data set is our Patch-seq data, which includes not only neuron morphology and electrophysiology, but also transcriptomic data. And you'll see on our lab tour in a few minutes exactly how that data is collected. But it's really almost sort of a Rosetta Stone of neuronal data, in that for a single cell, we can get all 3 of those modalities. And so we're inviting students to explore individual neurons in this data set by the transcriptomic type. So we introduced this idea of taxonomy of cell types, as the Allen Institute has been developing in our research in recent years, and then invite students to choose one of those transcriptomic types to explore the morphology of those cells. One tip you do really need students to be working on a printed copy of this worksheet so that they can actually sketch the neurons that they're observing. Could theoretically do it digitally as well if they're set up for that. But that's just sort of a practical consideration that we've found in testing this for students. With students, it's really useful to just have it printed out, give them a couple different colors of pens or colored pencils, and you get some really beautiful artistic sketches when you turn them loose with this. So again, some questions for them to explore about the morphology of the axons, dendrites, how they vary across cortical layers. I can't remember if I mentioned that all of this data is in this second data set is mouse visual cortex. So we have that caveat in the slides that all of these patterns that we're observing are true for mouse visual cortex specifically. And then again, some opportunity for partner or small group work to sort of compare one's findings. All right, hopefully you can see this now. So as I mentioned, this is meant to be really modular. You can choose to do one lesson or the other, but we do want to make sure that if you skip straight into lesson 2, that students have some familiarity with the concepts from lesson 1. So this slide just kind of summarizes that. And then you can see the histogram is, this is actually one of the concepts that confuse students the most in our pilot testing, which is why I am going to just really briefly address it. Essentially, it's just a representation of how much axon or dendrite there is in each cortical layer or arbitrary depth, distance in the cortex. So in lesson 2, this data set has the morphologies and the histograms overlaid on the actual cortical layers. But you may have noticed that in lesson 1, it's the same idea. It's the y-axis is just cortical depth. Cool. Any questions about lesson 2?
Claire, there's a question in the chat about if we have any resources for the hands-on laboratory protocols, I assume for either of these data sets.
Working on
Yeah.
answering that right now, so
Oh.
I'll link to
Wonderful.
some stuff. Yeah.
Okay.
Oh, great. Thank you.
That's awesome. Yeah, I think that's more in the realm of our scientific resources, because we're certainly not assuming that you will have the equipment to do Patch-seq yourselves as you are teaching these lessons. But certainly if you're interested in the scientific protocols and have the equipment to be able to collect your own data, that's fantastic. And I think in general, we're really excited to hear about when educators combine our open data sets with opportunities for students to collect their own data as well. Great question. So we're getting a question about how exactly we get this morphology data, and we're going to go into a lot more detail about that during the lab tour, but I will quickly show you. We actually have a little animation in the slides. This looks much cooler when you're actually showing it as a slideshow rather than scrolling through a PDF copy. But essentially the Patch-seq technique involves these are neurons in slice, either mouse or human tissue. And we combine patch clamp electrophysiology with a dye fill, a biocytin fill. And then so we can record, fill with the dye for later morphology reconstruction, and then also extract the nucleus for RNA-seq. And so that's where Patch-seq gets its name, the combination of patch clamp electrophysiology, RNA-seq, and then also our favorite in this lesson, morphology. Yeah, so great question about addressing the diversity of axonal complexity. Certainly, these are great resources for exploring exactly those kinds of questions. Our worksheets have students kind of compare general patterns in, sort of the axonal branching and anatomically where axons are going in excitatory versus inhibitory neurons and in different for lesson 1 and then for different transcriptomic cell types in lesson 2. But we're not currently really honing in on that specific aspect of it. But you certainly could use our data to do that. And that's also really helpful feedback as we think about continuing to revise these resources. And that is correct that Stacy or Rachel can elaborate on this a little more, but yes, this is in slice, so we don't necessarily have the full axon projection. So
Claire,
yeah,
I would just add that you're still going to get unipolar and not bipolar types of neurons simply because of the location for axonal diversity. Just wanted to throw that out there.
yeah, that is a good point that these data sets are not across the whole brain, they are cortex. So all right. I don't know if Stacy or Rachel wants to unmute and say anything further about that, but it is about time for our lab tour. So feel free to take a 30-second stretch break, continue exploring these websites, and I will see you momentarily outside of our lab.
Okay, awesome. Well, hi everyone. Welcome to the Integrated Cell Physiology Lab. My name is Megan Koch. I'm a Research Associate 1 on the in vitro single cell characterization team, and I'm going to walk you through our lab today. So we are just kind of getting set up. Everyone is starting to get patching. Some slices are on their way upstairs. Yeah, can everyone see and hear me all right?
I've got video and audio.
You got video and audio. Awesome.
Yep.
Perfect. All right. Well, we can start walking through a little bit. So here is the main area for IVSCC, which is the team that I'm on. So here we run our Patch-seq experiments every single day. Our main jobs here every day is just to run Patch-seq experiments all day long. So our goal is to collect the electrical, morphological, and then hopefully transcriptomic data through this experiment. So this is kind of like what our day-to-day looks like. So here is someone's rig all set up, ready to go. But I can walk you over to my station so we can get a little bit of a closer look. All right, so welcome to my rig. Here today, I have a slice of freshly prepared mouse tissue on my rig. So our goal is to try and get 5 cellular recordings every day from whatever slice we are given. So we work primarily with 3 species. We work with mouse tissue, we work with human tissue, and then we also work with non-human primates. So with that being said, where do we get this human and non-human primate tissue from? So we have partnerships with local hospitals and with the Primate Center at UW to get these tissues. So when they come to us, those tissues are still alive because we need live cells to record from. So we usually will get freshly prepared slices every day. So today we have mouse slices prepared. Now I can open this up so you guys can take a look at our slices. So today our focus is we're patching the nucleus accumbens and looking at ICJs, which it can be a little bit difficult sometimes because we jump between, not only we jump between species, but we also jump between regions of interest. So some days we'll be working in the caudate putamen and mouse, and other days we'll be working in the temporal cortex of human culture. So this is not the only type of tissue that we work with. This is what we call the acutely prepared tissue. And we get that from our lovely tissue processing team. They're awesome. But we also get culture slices. So those culture slices are sitting in an incubator in a media that feeds the tissue. So they can survive for up to 21 days, which is kind of incredible when you think about being able to keep someone's brain tissue alive for 3 weeks after their surgery, and it can be used that entire time, basically. So that's really, really fortunate. And with the culture, we can also apply viruses to those. So we can target specific cell types. That is our goal here, is we are trying to basically make a parts list of the human brain, what cells there are, what they do, what they look like, and how they function and their genetics. Yes, we have a question.
I was noticing they didn't see anything on the grids.
Yes.
Wondering how do you keep track of what slices?
Okay, yeah, so that is something that tissue processing will do for us. And I'm not gonna lie, it does get confusing at times. We do lose track of slices. But what happens is each mouse in the institute has its own ID. And from there, it will be dissected and sliced. And from there, tissue processing will take images of all of the slices. And each of these has a specific name and carousel. So each of those we call a carousel well. And they have corresponding carousel well names along with a cell ID and then a donor ID. So that donor ID belongs to the mouse. So we know what mouse it belongs to. It will have a slice ID. And then from there, we will also get cell IDs as well. So lots of numbers. We get these stickers every single day, and this tells us what we're doing. We also get morning briefs every day just to say, hey, this is the tissue we're working with today. So there are lots of numbers to keep track of, but thankfully we have something called LIMS, and LIMS keeps track of everything for us. Yep, management software. So thank goodness for management software. So I kind of covered all of the broad topics. Are there any more questions before I kind of get into a little bit more about my rig? All right, well, let's continue on. So I have my rig set up for today, and I have my mouse slice on here. So we are focusing today on, like I said, the nucleus accumbens. This is my microscope, and it allows me to control and watch the experiments as things are going on. And then, let's see, so this is all of the equipment that I use every single day. We have multiple programs running. So here's our scope. This is probably the most important and what makes this experiment unique. And this is what we call our workflow. So the IBSCC pipeline that we are in, we are trying to produce as much data as we can, and sometimes we can get a little lost. So we have this workflow that will help guide us through every single experiment. That is the cool thing about Patch-seq, is a lot of it is automated. And so that allows us to really ramp up production. Our goal is to try and get about 1,600 cells a year through our quality control metrics as well. So that is a lot, but the automation is really, really helpful in getting through all of this. Some of the other teams at the Institute do not have the automation that we do. And it is, you can see the amount of data that we produce every single day. So then, after kind of talking about this, I want to talk a little bit more about my rig. So these are specially created at the Institute by the Institute. And we have 8 of these machines. So this, I can do 1 patch at a time. They have that tiny, tiny glass pipette in there. And I have my mouse slice in my chamber at the moment. And so I also have fluorescent lights that I can check the labeling and expression of any enhancers or viruses that we might have applied. Trying to think what else cool stuff I have. Here are the amplifiers up here. And so today is kind of a little bit of a slow day just with mouse, but next week we have a non-human primate case coming up, which is really exciting. I think that's kind of what I have to show you around my side of the lab, but I can take you over to the multi-patch really quick and just take a quick look at that. All right, follow me. Okay, so the multi-patch is Patch-seq, but times a ton, times 10. So here is the multi-patch rig. You can patch up to 8 cells at a time with this, which is really cool because you can get an idea of more of like populations of cells and their activity rather than just looking at one single cell at a time. So using this, we can get an idea of how cells are interacting with each other within a given region. I just come and look at this every so often because I think it's really cool. No, I really want to work on it. I think our research associate too, Camillium, she's wonderful. I think she's done a little bit of work on this, but I think they're trying to automate this and add it into the pipeline. So hopefully one day we'll have some more people up and running on this. But for now, there is just one person rolling on all of these rigs. So got a question. How many members are there? Yeah, so we have 5 people within the lab working and patching every single day. We also have, there is one member from tissue processing that has learned how to patch. And so when we have an overload of cases, or if we just have really, really good slices, we'll bring him in for the day so that we can get as many recordings as possible. But there's 5 ladies in here and we have a lot of fun every day. Yeah. Mm-hmm. Yeah. Mm-hmm. Hands it off to you. Yes. What happens? Yeah. So after us, we put the slices into paraformaldehyde, and so then they get sent up to morphology. Because from there we want to visualize the cells that we patch. They get filled with biocytin dye, and so then they can get reacted with DAB, I believe, and from there we can visualize the cells. So those will go off to morphology, and then the nuclei that we collected, those will go off to transcriptomics. And eventually all of this data will get compiled and put back together, and that is that Patch-seq data set that you guys are working with. Mm-hmm. Getting a lot of excitement in the chat. Good, I'm glad. Patch-seq is really exciting.
We had a question earlier about when you're filling the cell, or when you're patching the cells, right? Are you filling them with dye, or what are you filling with them so that the morphology can be done later?
Yeah, great question. So along with so in those glass pipettes, we fill them with a biocytin with an Alexa fluorophore. So I think the colors are like purple and green. And so as we are patched into the cell, the solution inside of the pipette is diffusing through the cell, and hopefully is starting to fill it as you're recording. So I don't think that the more time you spend in a cell, I don't think that has an impact on how well the morphology is done or how much it has filled out. It depends on your seal. If you have a bad seal and you rip open your cell, you're going to lose your morphology because all of that dye is leaving the cell. So you want to do the best that you can to leave that cell, but keeping the membrane intact so all of the dye stays inside of that. And then once it gets reacted with that, sorry that I don't know the exact name for it, but I believe it's reacted with DAB upstairs in morphology. And from there, I think that allows us to be able to see the cells. I know that we have had issues with staining recently though. So. How long you typically spend with one cell? Good question. It depends on the cell type and the species too. So like chat cells are really, really big. Those can take up to half an hour, but sometimes you can be done recording a cell in 10 minutes or so. But I would say about that 10 minute to half an hour mark. I don't think I've had anything go past half an hour. Yeah. And with that being said, there's also extra time when you're setting up and looking for your cells. And then also the nucleus extraction takes a good amount of time. So what we do is once we are done recording the cell, we then apply negative pressure to that cell and wait for about 4 minutes. So that's already adding 4 to 5 minutes onto your recording. And then from there, it's going to take another 4 minutes to extract the cell because you're basically trying to move as slow as you possibly can while maintaining a seal. So there's so many aspects that we're having to control and keep track of all at one time, but you get into the muscle memory of it, and it gets a lot easier. And you watch people and they're patching, but they're looking away and talking to you. So I think we have a really great group dynamic going on, and we have a lot of fun in here.
Awesome. Well, if there are any last questions, go for it,
Mm-hmm.
and then we'll take you over to.
Mm-hmm.
You're getting lots of appreciation in the chat.
Wonderful. Well, I'm glad you all appreciate Patch-seq. Yeah. Well, I'm glad that I got to show you guys around today, but I got to get back to patching. So have fun upstairs.
So after Patch-seq gets sent up to the imaging lab, we also, of course, have an automated high throughput process for imaging all of those. And then the images get sent to our morphology team down here. So we're going to just take a look at how Ingrid and the other folks who do all that reconstruction do their work.
Hi, everyone. So welcome to our morphology area. So over here on my computer is our images that we get from our Patch-seq team. So over here on the left side is a 20x image. So the cell that we're specifically looking at is cell 5 over here. So this gives us a really large scale idea of what the image looks like. So up here in the orange is our PIA, and then down here in the teal is the white matter. And then we can see each of the 6 layers are drawn. So as I mentioned, the cell that we're looking at today is cell 5, which is located in layer 3. And this is specifically a human cell that is LAM5, and it is in the middle frontal gyrus. So over here on the right side is VAW3D. VAW3D is our software that we use for reconstructing. So right now we're viewing a 63x image of the cell. And this is an overview image. So this combines all of our 2D images, but if we go into our polyline function, we can scroll through each of those 2D images and see kind of how the cell develops. And then I can add on our reconstruction. So this is a large overview of it, but we can also zoom in on our resolution and get much more zoomed in all the way to the soma. And we can scroll through and we can see how the axon starts to branch out and the dendrite. And if we zoom out, I can orient you all through what all these different colors mean. So at the top, we have pink. Pink is our apical dendrite. So we're going to see that that's extending from the soma out to the cortex. And if we zoom into the soma, we can see that the apical dendrite's a lot thicker than the rest of the dendrite, and it creates one large branch that then has multiple branches that come off of it. And then over here in this navy blue is our basal dendrite. Most of it branches at about a 90-degree angle from our apical dendrite. And it kind of follows a similar pattern, but we can see that in contrast to our apical dendrite that only has one branch from the soma, we have multiple basal dendrites. And if we zoom through all of our 2D images, you can see that they all come out at different points. And then lastly, which I think is the coolest, is our axon. Axon is marked in red, and typically it comes out from the soma only, well, it only comes out at one point from the soma, and usually at about a 180-degree angle from the apical dendrite, but in this case, we can see it's a little bit less than that. And if we zoom in, we can see right here is the axon that's coming out. And one of the things that's really cool about axon is it's a lot thinner. So if we go into our reconstruction, we can see right here, this is the axon that's coming out. It's a lot thinner. And if we zoom out, we can see that also the axon has a crazy pattern compared to the rest of the dendrite that we have. So typically dendrite, we see that it follows a very uniform pattern with acute angles, but with axon, we can see that it's kind of crazy. It goes all over the place. Down here, we see some like 90-degree angles. We see some sharp turns around here. We also see it even turn back towards the soma. As far as our actual reconstructing process, I can zoom in and show you kind of what it would look like. Just crashed. That's okay. You can go ahead and open back our image stack. So what I'm doing right now is I just opened up our software again, and then I'm opening up our resolution folder. I'm going to orient that, change some of the coloring, and then I'm going to go ahead and open up my annotation file. You can zoom back in. So typically when I'm reconstructing, one of the things I do is I'll start at the soma and mark the soma. And then I like to start at each branch from the soma and I'll start reconstructing. And then I like to mark in like a teal blue, as we can see up here, each of the fork points. And then I'll kind of work out from the soma towards the end of the dendrite and trace each of those points. So what we're going to do is we're going to come over here and we're going to trace this branch. So what I'm doing right now is I'm zooming through each of our 2D images from what we're viewing right now, which is the top of the plane to the bottom of the plane until I see the signal come into focus. And I can make the annotation appear and disappear. We'll do it like this. And then I'm going to change the type to apical dendrite. And I'm just going to be scrolling through the Z plane and marking it as it comes into focus. Just like that. And you can see it's creating each of these nodes that then when I go off of our Z-plane, then it creates a reconstruction and I'll attach that together. And then keep on going. You can zoom in even more sometimes. This allows you to see it a little bit better. And again, I'm just scrolling until I see my signal. And I'm just going to follow the signal right along there. Just like that, and then I'll join them both together. And so the idea behind this is once we trace all these branches, then we're able to get a 3D reconstruction of the entire neuron. And you can kind of view it like this. And then we get a full reconstruction.
Hey, Ingrid, we have a couple questions in the chat.
Yeah.
One of them is there a second level verification by another scientist as part of the process of mapping?
Yeah, so specifically on our morphology team, we have 3 research associates. And so for each person that works on a reconstruction, we'll go ahead and we'll do the reconstruction ourselves. And then we have someone else from our team go through and they'll do a tag team process. So essentially what they're doing is they're going through each of our reconstructions and making sure that they agree with all the branching and all the connections that we make. So yeah, everything is proofed by another scientist. And then even once we upload the reconstruction, we have software that kind of goes through and manually checks our branching because we'll do radius correction to get the thickness of the dendrite branch as well.
Great, thank you for sharing that.
Yeah. So that's another look at it. And then if we turn over here to the right, we have another member from our team. So you can see that this looks a lot different than the reconstruction that I was doing. So this individual, Natalie, she is reconstructing in VR. So this is another way that we can visualize neurons as well. This is really helpful for reconstructions that we're doing that use other techniques, even outside of Patch-seq, because it allows us to see how neurons extend in 3D space across further distances.
We had some good questions in the chat further up about sort of how far you can trace these neurons in slice and whether we have other data sets that allow you to follow the axon further distances. And the answer is yes. Definitely check out the Connectivity Atlas. And then our Neural Dynamics Institute has some really exciting data coming online from their new exospin technology, which is a light sheet microscope combined with clearing and expanding whole mouse brains to be able to reconstruct neurons wherever they're going in the brain. And so this is what you're seeing here with the VR tracing would be the sort of technique that's used for those reconstructions. Any other questions for Ingrid? Question about why the headphones? I assume referring to the VR tracing? Yeah. Ingrid, do you know why headphones are built into the VR headset, or I'm not sure that they're
You
actually being
know,
used.
yeah, they're not being used. I believe that's just the headset. I don't work specifically on our VR, but yeah, they're not being used.
Cool. Awesome. All right. Well, thanks so much, Ingrid. Thank you, Stephen, behind the camera. And I think we'll spend a couple minutes taking any other questions that you have for Ingrid or Stacy or Rachel about sort of the Patch-seq technique and the data sets that these lessons make use of. Yeah, some good questions about Patch-seq. I don't know if Stacy or Rachel, you can comment on that, but my understanding is that because the reconstruction relies on this biosite fill, if that's leaking out, you're not going to be able to do a reconstruction because you can't visualize the morphology of the cell.
Yeah. I would also add, there's the question about, do you reconstruct with a hole? Oh, just the cell reseals after extracting the nucleus. Otherwise, the biosign wouldn't be contained in it. So.
Yeah, but it is a good point that if it had spread through the branches, maybe it would actually be okay. But we find that if we don't get that reseal, then it just doesn't, the cell looks bad, So maybe if we had nicely intact dendrites and axons, we would feel like we could, miss out on that soma, but that's not how we do it. It's true that we don't do a lot with the soma morphology with Patch-seq because of the fact that we, one, patch the soma in the first place, but also because we pulled that nucleus out. And so we don't feel like we're getting a good representation of what it would look like normally. So
there's a question about VR tracing versus what Ingrid was doing. Do you want to speak to like the different uses for that, or they're really based on different data sets? So we only use what Ingrid, that was really Patch-seq based. So when we're reconstructing in a slice, it's got some nice tools that allow you to basically see plane by plane instead of seeing everything in 3D, which is very nice for slice. But the VR has that's for our whole mouse brain morphology. So that's when you're navigating an entire mouse brain, and it has added benefits to be in that 3D space in VR. Yeah, Stacy, you got anything to add to that?
Well, the only thing I would say is that I know they like it in particular for so with the whole brain data, we have multiple neurons labeled, and so one of the most important things that we have to do when we reconstruct those neurons is to make sure that you don't confuse the branches of one neuron with another one. And so when they coalesce together, having that three-dimensional view of their trajectories helps to just decide which path you should follow. And so yeah, and then people that the reconstructors like Natalie say they feel like they can see the neuron better. So but we feel like we can see it pretty well in the slice with the other approach. So yeah.
So there's a great question about whether an educator could have a student perform VR tracing. This is a good opportunity to just quickly mention that eagle-eyed observers may have noticed in the old version of Neurons Beyond the Textbook, we had a homework assignment that used Mosaic, which was a citizen science neuron reconstruction website, which sadly is now defunct. So we have taken that out of our lessons. I would be really curious to know, just by, comment in the chat, if that's something that any of you have used before and found valuable or think you would find valuable, because while we don't have control over the fate of Mosaic, that is something that we really want to find a way to let educators do this kind of neuron reconstruction in their classrooms, whether in VR or just on a laptop screen, the way students could in Mosaic. Yeah, go for it, Stacy.
Okay. Yeah. So I just wanted to say, because Claire, you and I have never talked about this before, but basically because of the reconstruction pipeline that AIND is setting up, which Abe's Brain Science will also be a big part of it, especially a user of it. That is an online reconstruction platform. It's the whole brain data, but it would be very cool to send people there and have them do similar things that they did in Mosaic in the Horta space. And so that is the place where I think, like, someone asked, could we have our students reconstruct in VR? Because that would be really cool. That is not currently in the works, but that is a possibility to add VR to this cloud-based reconstruction platform. And so if there was, a big interest in it and the use case, then we could probably push that. But that does seem like it could be pretty cool to do that. So
And I'll just, oh, I'm sorry, Stacy, go ahead.
go.
I was just going to say, I'm going to just take a moment to plug. After this workshop, you will receive a feedback survey, and that is exactly the kind of information that we would love for you to share, what we can do to provide more useful resources and tools for you all. Because if we hear enough interest, potentially we can make things happen. Okay, I want to make sure that we're staying on track time-wise. So I'm going to just very quickly share my screen and let you know what we're going to be doing in breakout rooms. So we're going to take about 10 minutes, maybe closer to 8 at this point, to put you all in small groups and give you an opportunity to reflect a little on what you've heard today. Think about how you might implement these lessons in your classes. What would be the next steps toward that? What obstacles or challenges you might encounter, and what resources we might be able to provide to help you overcome them. Welcome back, everyone. So I know we only have 5 minutes left. Rachel's going to give us a really quick summary of sort of what we've observed on the mural board. Thank you all so much for taking notes there.
Yeah, we really appreciate it. So I've been looking at the mural board, and it looks like the classes that y'all are hoping to implement these in are things like the morphology part of Intro to Neuroscience, maybe some more upper level neurobiology slash like morphology classes. I saw a data analysis course, potentially an online cure. I saw a bioinformatics course as well. And so there is there are some things kind of outside of Intro to Neuro, which is in my mind like where I've implemented the previous version of this before. Um, the next steps towards implementation, it looks like there is, it's going through the materials, seeing how they would fit in class, maybe doing some adaptations to make it work for your class. So yeah. And then the obstacles and challenges. Right, there's a big one. It's teaching methods is difficult, which I completely agree with. And so figuring out a way to teach the methods, right, along with kind of the outcomes for what the neurons look like, right? And finding time, fitting it into a curriculum, especially if you have a content heavy, curriculum or plan for your semester, that can make it trickier to fit in something that's very active like this. And large class sizes, right, are that's often right, a sticking point with things because there's just a lot to manage and a lot to manage, especially with flipped classroom if you're doing that with the assignments. So yeah. And then I'm seeing that y'all are considering resources to help overcome, right? Um, right. So maybe using group assessment. I know that actually works really well with large class sizes. If you have TAs, right, to help with this. Um, ooh, Having that VR tracing learning module would be helpful. Um, and yeah, so thank you so much for contributing on this. We really appreciate it. And I hope y'all should be able to see each other's and if there's more resources that show up, you'll be able to see them. We're also going to take this info and hopefully address some of these in the instructor guide. That as it's being completely finished, right? We can see the obstacles and we can maybe provide potential solutions for them, or at least ideas to think about them. So yeah.
Awesome. Yeah, we're as Rachel mentioned, these materials are still a work in progress. You all have gotten a sneak preview and gotten to see some drafts, but particularly those instructor guides, we are going to flesh out some more, provide some suggestions for, different ideas for remixing and hopefully address some of the potential obstacles and challenges that you've raised today. Feel free to keep adding more thoughts to the mural board, and then that brings me to my next point, which is we have a feedback survey that we would love to get more of your input on, both about the curriculum materials, but also about this workshop. We had the wonderful problem that so many of you were interested in this, that we couldn't make it quite as discussion-based the whole time as we might have otherwise. So we would love to hear from you how that went, and what we might do similarly or differently in future workshops. What would be most helpful to you? We're also going to have an opportunity, if any of you are interested in being a pilot tester for these lessons. So we're planning to have final for now versions up on the website publicly available later this month. But then we plan to keep iterating on them based on feedback from educators like yourselves. And when I send a follow-up email next week sharing a reminder about the survey, a link to the recording of this workshop, I will also have a sign-up form where you can express interest if you would like to be a pilot tester and more formally give us some feedback in exchange for just a small incentive about how these materials are working in your classroom. And lastly, this came up earlier, but we do have a summer internship program for undergraduate students. Please spread the word. You can learn more about that and our postbac program and research associate positions like Megan and Ingrid and some of the other folks you met today, all at alleninstitute.org/careers. I know we are a minute over, but I'm really quickly going to turn it over to Rachel to just mention some other really exciting opportunities coming up next summer.
Yeah, so we have some faculty development opportunities for next summer. The first to kind of kick off our series is that we are going to be holding an open science and undergraduate education symposium in June. There will be more information about this released soon. If you're on our educational newsletter, you'll get information from there. We can also send out the info to the list of folks who signed up for this event. We'll have multiple workshops going through July and the beginning of August. We're going to have a coding with Allen Institute data, basically a Python workshop for those of you that might want to start integrating programmatic access to the data sets into your classrooms as well. Developing open neuroscience teaching tools is a activity and course design workshop. Basically, we're going to go through some of the data sets and work collaboratively in small groups using backwards design to design some resources, right? And that one will have a year-long community of practice that goes with it as well. And then in the beginning of August, we'll have Teach Extravaganza, which is a cell biology collaborative biology curriculum development workshop, which is also available. So we
So
hope that
stay tuned for more.
yes, please stay tuned for more. And we're hoping we're going to see y'all at these in the summer.
And with that, thank you all so much for your time and attention and all your great discussion in the chat. Here's our website. Here is the Allen Institute for Brain Science data portal again. Please feel free to stick around, continue to put any questions, comments, thoughts in the chat. All of us here at Allen will be around for a few more minutes to take any remaining questions. So thanks so much.